Thank you very much. First of all, I want to be very thankful for this invitation. It's really a pleasure to be here for me. It's my first time in Russia, and so far I'm enjoying it very much, so thank you. I come from Public Health Institute in Valencia, in Spain, where we look at several different areas that have to do with biomedicine and public health. And in particular, in my department, we work with genomics and health issues, mostly related to bacterial genomics. And a lot of us do work that has to do with understanding the human microbiome. So this is the first talk for this meeting about human microbiome, so I'll give a very brief introduction. Basically, when we talk about the human microbiome, what we are referring to is the large collection of microbes that inhabit the human body. So we have a large number of bacteria that live on us and within us. A typical healthy human probably has in the order of 10 to the 14 or 10 to the 15 bacterial cells, which is obviously a very, very large number. And most of these microbes are not in transit, but actually inhabit defined niches in our body. So we have specific adapted microbial communities that live in different niches of our body, such as the nasal cavity, the oral cavity, the skin, the gastrointestinal system, or the urogenital system, for instance. Those would be the most important. This is nothing special about our biology. This is very common in nature in general. In fact, bacterial communities are found in association with all kinds of living organisms in our planet. So vertebrates, invertebrates, plants, marine systems, every multicellular organism in our planet lives in association with microbial communities of some kind. And this shouldn't be surprising at all. In fact, it should be totally expected, because if we think about the evolution of our planet, clearly for many millions of years, billions actually, there was nothing on our planet but bacterial cells. When the first eukaryotes appeared, which would be right here, they appeared in a planet that was already completely covered by microbes. So it's only to be expected that every eukaryote has had to evolve in this context and has evolved as a community with microbes. What I'm going to talk about today is specifically the human gut microbiome, that is the microbes that live in mainly in the intestines, and actually mainly in the intestinal lumen. So this would represent, in this picture, this would be the epithelium of the intestine. Then we would have a layer of mucus that separates the bacteria that are densely populating the lumen from this epithelium. Although we also have some bacteria that may live in the mucus, or even some of them in contact with the mucosa. The human gut microbiome is the niche, the microbial niche that has received the most attention in human microbiome studies, and there's several reasons for it. For starters, it's a very, very dense community. It's actually thought to be the community with the highest cell density of any known ecosystem on Earth. So we're talking about 10 to the 11 or to 10 to the 12 cells per gram of luminol content in the large intestine. The number of cells is, as we said before, very large. So our entire body is supposed to have around 10 to the 13 bacterial cells, and so the microbial cells that we carry probably outnumber the human eukaryotic cells. It's very diverse. It includes hundreds of different bacterial species, also archaea, viruses, and some eukaryotes, but mainly, or at least what we have focused on the most, is the bacterial species, and many of them are still uncultured. Another aspect that is very interesting about the human gut microbiome, well, interesting and intriguing, is that there is very large variability among individuals in terms of age, diet, health status, geography, lifestyle. There are many factors that influence the composition of the gut microbiome, and we're just beginning to understand how this happens, what originates or what regulates this variability. But ultimately, of course, the reason that we care a lot about the human gut microbiome is because it performs a lot of very important functions for us as the host. Some of these have to do with nutrition and metabolism. So for instance, it is thought that about 10% of the energy that we extract from food is actually extracted by the bacteria in our gut from complex carbohydrates that our own human enzymes would not be able to digest. Another very important function is related to protection from pathogens. So the fact that we have this very dense and well-adapted bacterial community in our gut protects us from potential infections by pathogens, by competition, by production of antibacterial molecules, etc. The microbiota also has a very direct interaction with the gut tissues, and is actually involved in regulating the development of these tissues. And last but not least, probably the most important of the functions of the microbiome is the one that has to do with its interaction with the immune system and its very strong role in sort of guiding the development and the modulation of the immune system, especially in infants. So the gut microbiota produces or contains a lot of different molecules that end up interacting with many very important physiological processes of the host. So this is just a large view summary of some of these interactions and some of these molecules. So for instance, the microbiota produces short-chain fatty acids. It transforms primary bile acids to secondary bile acids. It produces indoles, hormones, and neurotransmitters that end up interacting with many different processes that can go from immunity to lipid homeostasis, glucose homeostasis, communication with other tissues, the brain, for instance. A lot of research is going on these days on the communication between the microbiome and the gut-brain axis, etc. So basically, I put this up here to insist on the large variety of processes that the microbiota interacts with, and also the large overlap that there is in all of these interactions. An important thing to keep in mind is that we are realizing that our current lifestyle is affecting the composition of our microbiome in comparison to what our ancestors likely had in many ways. Probably some of the main things that are responsible for these alterations are, on one hand, dietary changes, of course, also everything that affects our exposure to microbes, such as the improvements in hygiene that we have in our societies, and of course, the very large use of antibiotics that we do. So this will affect especially the development of the microbiome during childhood, and it can be related to several health problems. For instance, through the effects that an unbalanced microbiota can have on immune development, we have that children can be more prone to having atopic diseases, or through the effects that it can have on metabolism. We can see that an altered microbiota has also been related to metabolic problems such as obesity and the metabolic syndrome. More recently, there's also evidence that through the effect that an unbalanced microbiota can have on neurological development through unbalanced production of cytokines and neurotransmitter molecules, it can, of course, have to do with the development of, for instance, diseases of the autism spectrum. So in this sense, it is very important to try to understand as well as we can how the gut microbiota develops during infancy and childhood, so that we can learn eventually to try to modulate this process when necessary. And this is what we do in my group through mainly a series of different omic analyses. And today I'm going to talk basically about the development during the first year of life, so from birth to 1 year of age, which is the period during which most of the development of the microbiota occurs. So it's a period in which we go from a gut that is probably not completely sterile, as we used to think at birth, but at most populated by very few microbes, to a community by 1 year, which is already very complex and that can be comparable to that of the adult, although not yet identical. So this first year of life, we have studied through analysis of a small cohort of babies born in Valencia that we analyzed, from which we obtained fecal samples, as well as food questionnaires, among other metadata, and for which we've applied a series of omic analyses. And what I'm going to describe today is based on metagenomic and metatranscriptomic analysis. I should also point out that for every one of these babies that we follow through this first year of life, we also obtain samples from the mothers, and we obtain samples right before birth, and we also obtain samples 1 year after birth. And most of what I'm going to present today has actually already been published. The metagenomic analysis were published a few years ago in PLOS Genetics, and then earlier this year we published the metatranscriptomic results, obviously in comparison to the metagenome results. So basically, as I said, most of what we do is based on omics analysis, and this is related to the fact that, as I said at the very beginning, there is still many bacteria in the gut microbiome that cannot be cultured, and therefore omics analysis give us the opportunity to look at these bacteria, because basically what we're doing is obtaining total DNA or total RNA from these communities without any attempt of separating bacteria or culturing them. And then we work from this total DNA or total RNA. If we work from total DNA, we call it metagenomics. If we work from total RNA, we call it metatranscriptomics. So very briefly, I'll go through some of the main results from the metagenomic analysis. Basically, what we are plotting here is the composition of the microbiota in the fecal samples of these infants and their mothers at the different time points that we analyzed. So we have I1, I2, I3, I4, and I5 are the 5 time points which we study for the infants, which I'll remind you correspond to 1 week, 1 month, 3 months, 7 months, and 1 year. And then MA and MB are the mother before and after birth. And so basically what I want to point out here is that if we look at this in a correspondence analysis plot, we can see how even though the dots that correspond to each one of the samples are not perfectly separated by time point, we do see clearly a movement through time from the composition of the youngest babies towards that of the mothers, both in terms of the taxa that we identify in the metagenomic sequencing and in terms of the functions that we identify. Here, this is a similar plot, but what we have done here is show the space in the plot that each one of these samples occupy, that the points corresponding to each one of these samples occupy, and this allows us to show how there is much more inter-individual variability at the beginning, and then this reduces as the baby grows old, and in adults the variation between individuals, even though we know it's very large, is much smaller than what we have in younger babies for taxonomy, and again also for function. Another thing that we did with our metagenomic results was to try to look at the potential interactions between the taxa that we identified, and we did this through analysis of networks of co-occurrence among taxa. And this is summarized here. So this figure is a little complex, but I think it gives some interesting information. So what we have here actually is the network based on the taxa of the adult, okay? The taxa of the mother, the core taxa that were present in all of the mothers in our study. And then what we have done is we have colored these potential interactions based on the time point in the baby in which they appeared, okay? So the ones that are, these few ones that are colored in red here are interactions that were already present in the infant at 1 week. In blue, the interactions that appeared at 1 month, etc. So E4, E5 in purple and orange are the ones that appeared at 7 months and 1 year. And we can see that at this point, we are already obtaining a very dense core of the network with a lot of interrelationships. And then these brown lines here would represent the interactions that are present in the mother, but that we did not identify in the infants. And so interestingly, we can see that all of them are in the peripheral area of this network. So basically what we interpret from this is that by 1 year we already had the core of the network in place. We've also been looking more recently at networks of metabolic interactions, trying to take into account the data that we have from the food questionnaires. Okay, so we've been for these ones in particular, we have used the data that we had from metagenomics, and we have translated the information that we had in our food frequency questionnaires to a list of input nutrients that includes minerals, carbohydrates, amino acids, vitamins, lipids, fibers, and flavonoids. And then we've used these two types of data to create a supraorganismal metabolic network that is not looking at individual metabolic networks, but trying to understand all of the possible metabolic reactions that could occur within the community. And what we obtain is what we see here. So basically, I guess the main overall result that stands out is that we find when we look at the reactions that we find in the different networks corresponding to the different time periods in the infant, we see a very big jump between the time points that correspond to lactation and the points that come after the introduction of solid foods. This is less apparent when we just look at the metagenomic reads, and it becomes much more apparent when we look at the reactions within our metabolic networks and the metabolites that are produced. So we wanted to see which were the metabolic networks that changed the most actually before and after the introduction of solid diet, and this would be the top 10 metabolic networks for which the dissimilarity score was highest. So one of the things that stand out is, for instance, the metabolism of phenolic compounds, so things such as flavonoid biosynthesis and phenylpropanoid biosynthesis are almost absent in milk feeding infants and become much more abundant after the introduction of solid foods. In reverse, for instance, things such as vitamin B6 biosynthesis are more common during the milk feeding period and then decrease after the introduction of solid foods. We looked experimentally at trying to confirm some of the results that we got from the metabolic networks, and we did this for a phenolic metabolite called ferrylate that is known to be related to the gut-brain axis and to have some neuroprotective capacities. And we found that indeed experimentally we could detect a large increase in the production of this metabolite between 3 months and 7 months, and then no significant difference between 7 months and 1 year. So basically this is jumping here with the introduction of solid foods. And we could also relate that to the amount of the nutrients that we know from our networks that our networks are predicting to be involved in the synthesis of ferulate. So if we go to our food frequency questionnaires, we can also clearly see that these nutrients appeared at this point. Now for the last part of the talk, I want to switch to what we've learned not from genomics, but from metatranscriptomics, which is what we've done more recently. So for metatranscriptomics, if one is interested in learning about the genes that are being expressed at a certain point in time, there is one first experimental step that one has to do before sequencing, which is removing ribosomal RNA. This is because the immense majority of RNA that is present in a cell is actually ribosomal RNA. Therefore, if we sequence total RNA directly, most of what we're going to get are ribosomal RNA sequences, which can be informative, but do not tell us about what genes are being expressed. So since that is what we wanted to do here, the first step was the removal of RNA, and then of course from that you synthesize cDNA and you sequence it more or less in the same way that you would sequence DNA. So I'm going to be relating the results to what we see in the metagenome. So the main categories that are most abundant in the metagenome and the metatranscriptome are similar. So basically things that have to do with carbohydrate metabolism, with protein synthesis. But one thing that we see here as overabundant in the metatranscriptome that we did not see in the metagenome are proteins that have to do with protein folding and protein stabilization. So that's one of the main differences between metagenome and metatranscriptome. If we look at the progression of the metatranscriptome through time, so here I would be comparing, this would be similar to the plots I showed you before, except here we'll be comparing what we see at the metatranscriptome level and what we see at the metagenome level. We can see that again we have fairly clear progression from the one we called infant towards what we see in the adult. If any, actually better or more clearly seen in the metatranscriptome actually than in the metagenome. We can employ different techniques to try to define sets of functions that follow similar paths through time, both in the metatranscriptome and the metagenome. And we can define functions that tend to go down during the first year and functions that tend to go up. Some of these coincide, like clearly things that have to do with aerobic metabolism go down in both. And we have also compared the differences that remain between the infant's metatranscriptome and the mother's metatranscriptome 1 year after birth. And we can see that there are some functions that are more abundant in the metatranscriptome of infants. For instance, again, those related to aerobic metabolism, even though they go down throughout the first year, they're still more abundant in the 1 year old that they are in the mothers, and also those that have to do with protein synthesis. So maybe suggesting that bacteria in infants are growing more actively. On the other hand, functions related to central intermediary metabolism, methanogenesis, lipid degradation, protein interactions, are still less abundant in 1-year-old infants than they will be in mothers. Now, one very interesting, at least to us, thing that we've noticed in our data set is that if we compare the functions that we detect in the metatranscriptome of the samples that we have from the mothers before birth and 1 year after birth, we detect some significant differences, and notably these differences are almost the same that we detect between the mothers 1 year after birth and the 1-year-olds. So again, functions related to aerobic metabolism are more common in late pregnancy than in mothers, and all of these functions that were not seen in 1-year-old infants, such as methanogenesis, lipid degradation, some functions related to protein interactions, we also see them diminished in the mother during the end of the pregnancy period. And I'm going to go, I'm going to comment on that a little bit later, but first of all, I want to show the main type of analysis that we have done with our data, which is not looking at the metatranscriptome by itself, but actually relating it to the metagenome data that we have for the same samples, and therefore defining which genes are under or overexpressed in relation to their abundance in the metagenome. And so basically, in this heat map, we are showing in blue the genes that are underexpressed in the metatranscriptome, and in red the genes that are overexpressed. So first, the first thing that I want to show is that we find that the samples from the different time points that we've analyzed cluster basically into groups. On one hand, we have the younger infants up to 7 months of age, whereas the 1-year-olds already cluster with the mothers at point A and point B. And again, that the infants and the mothers at late pregnancy are actually more similar to each other than they are to the mothers at point B, so 1 year after birth. The other thing that is clear from this plot is that many more functions are underexpressed than overexpressed in general over all time points. In fact, there is only these 4 categories here that are systematically overexpressed. That would correspond to things that have to do with glycolysis and gluconeogenesis, things that have to do with synthesis and modification of ribosomal proteins and with translation factors, and as I already mentioned before when we looked at the first heat maps, things that have to do with protein folding and stabilization. On the other hand, a large number of functions are systematically underexpressed, so in blue here. These would be things that have to do regarding energy metabolism, things related to the metabolism of amino acids and amines, that's underexpressed overall. And also things that have to do with tRNA amino acylation and protein modification and repair. Other categories that are systematically underexpressed would be things that have to do, again, with this time transport of amino acids, peptides, and amines, DNA metabolism, things that have to do with prophages, always underexpressed, things that have to do with sporulation and germination, and toxin production and resistance, hopefully always underexpressed, and also many proteins that have to do with biosynthesis of a variety of different compounds. However, maybe the more interesting information that we obtained from this analysis are not the genes that are always under or overexpressed, but those that actually depend on the stage at which we are. And we do find quite a bit of those. So for instance, we find, again, that everything that has to do with oxidative phosphorylation and protection from oxidative stress is overexpressed in young infants, but then becomes underexpressed later on. And on the contrary, things that have to do with biosynthesis and degradation of polysaccharides, transport and binding of carbohydrates, and phosphotransferase system tend to be underexpressed in young infants, but become strongly overexpressed later on. So this is overall in accordance with what we know of the physiological changes during infancy, so decrease in oxygen tension and increase in the variety and complexity of the dietary carbohydrates that the infant is going to intake. One thing that we didn't expect is that we detected that some enzymes that are related with the synthesis of butyrate are significantly overexpressed at 3 months of age. So at this age, the bacteria that usually synthesize butyrate in adults are not yet present or are not abundant, and it was thought that maybe butyrate production was not as important for milk-feeding infants as it is known to be for adults. But we are seeing that, in fact, we can detect an overexpression of butyrate-producing genes specifically at this time period, and specifically in Clostridium species that are not the typical producers of butyrate in the adult. Finally, again, if we look at the differences between infants and mothers, we detect, as we saw already when we were looking only at the metatranscriptome without relating to the metagenome, that there is a certain series of functions that have to do with methanogenesis, degradation of proteins, biosynthesis of natural products, and some regulatory protein interactions that are underexpressed in all infants but overexpressed in mothers. So clearly, the maternal microbiota is more complex, has a more diversified metabolism, possibly with a more sophisticated regulation than what has been attained by 1 year of life. And well, this reinforces the notion that the microbiota succession process is not yet complete at 1 year of age. So if we summarize all of what I've said in this figure, together with what we know about changes in bacterial composition during time, some of the things that we can highlight is that so both gene abundance and gene expression change through time, not necessarily in a completely parallel way. There are things that we see when we look at the expression that we don't see when we look at gene abundance. For instance, if we look at, in one we called infants, if we look at gene abundance, the ones that are that stand out in particular at this period would be things that have to do with the TCA cycle, of course, PTS, degradation of milk sugars, whereas if we look at gene expression, we detect a lot of genes expressed that have to do with protection from oxidative stress, and also with glycolysis and gluconeogenesis. During the milk feeding months, the most abundant genes in the metagenome would be lactose transporters and things related to the degradation of the sugars present in milk. Again, at the metatranscriptome level. We also see enzymes related to carbohydrate degradation, but we also detect this butyrate synthesis from Clostridium that was kind of unexpected. If we go to older infants that are already eating solid foods, we detect a metagenome largely already similar to that of the adult, but with a main difference relating to methanogenesis functions which are not yet present in the infants. And the same we see in this case at the gene expression level. Another thing that is remarkable is that, as I mentioned before, if we look at what genes are underexpressed or overexpressed in mothers before giving birth and mothers 1 year after, again we detect that these genes that differ are, or some of them are the same that we were detecting when we were comparing 1-year-old infants and their mothers 1 year after birth. So methanogenesis, regulatory protein interactions, degradation of proteins, peptides and glycopeptides, and biosynthesis of many natural products are also underexpressed in late pregnancy. So these may suggest that these late coming functions that appear after 1 year of age may be less central to the functioning of the gut ecosystem and get also downgraded in very specific physiological situations such as those of late pregnancy. In addition to these genes, we also detect some other differences between the under or overexpression of genes in late pregnancy as compared to 1 year after birth. For instance, there are many genes that are normally overexpressed that relate to biosynthesis and degradation of polysaccharides, synthesis of DNA replication precursors, and carbohydrate transport systems that we no longer see being overexpressed during late pregnancy. And actually the only genes that we see overexpressed in the late pregnancy samples are a PTS transporter that is actually special as PTS systems go, in that is fairly general for different types of sugars and can actually internalize glucose. And most remarkably maybe, genes that have to do with synthesis of glycogen. These we only see overexpressed in late pregnancy. And at the same time, some genes that are usually underexpressed are not underexpressed at this period. So things that have to do with stationary phase and with different types of stress, such as heat shock and phase shock, are normally underexpressed, but not at this point. So this is totally speculative at this point, but we think that this simplified metabolism that we see in the microbiota of late pregnancy appears to be enabling the microbiota to use an excess of glucose in the environment. So everything that has to do with complex carbohydrates that is normally in function in the adult microbiota gets shut down, and all we are expressing is a glucose transporter and glycogen synthesis. So it would seem that somehow these bacteria are having access to an excess of glucose, which of course pregnant women do have an excess of glucose in their blood. Whether this should be available to the microbiota or not is not so obvious, but this data would suggest that it is. The other speculation at this point that we can have is that maybe these bacteria are in stationary phase, basically due to this lack of underexpression of these genes. That could also be very relevant because, of course, that means that these bacteria would be in a very different physiological condition than they might be in other time periods, and that may be related to their capacity or not to translocate through the epithelial membrane, which is something that is of relevance, especially in late pregnancy, because in late pregnancy the gut barrier is somewhat altered. There is more possibility of translocation of bacteria at this period, and so the fact that these bacteria might be in stationary phase may affect the possibility of translocating. So just to summarize and conclude, basically what I've been telling you today is that in spite of the large variability that there is in the individual patterns of gut colonization in infants, we can still detect some general trends. These trends clearly show a directional change towards the taxonomic composition, functional composition, and also gene expression patterns of the adult microbiome. The trends that we see at the level of gene expression are not necessarily identical to those that we see when we look at gene abundance, because the relative expression of genes may change through microbiota development. So we're detecting some stage-specific metabolic adaptations, some specificities related to late pregnancy, and others to physiological changes throughout the first year of life. And again, that by the last time point that we explored in this analysis, which is 1 year of age, we can still see important differences between the microbiota present in these infants and that we see in adults. So clearly there are still changes that will have to occur during childhood and adolescence, and that is something that we are exploring in other projects in the lab. And to end, just remind you that basically if we are interested in these, it's because probably these first months of life and also the period of pregnancy may be some of the most important periods in which interventions that are aimed at modulating the gut microbiome may be most likely to succeed. And more consequential for health throughout life. So it would be very important to learn to modulate the microbiome during this period, and this of course means that we need to understand very well how it is normally developing in healthy infants. And I will finish here. I would like to acknowledge people in my group that have participated in this work, notably my postdoc Yvonne Vallès, who is now a professor in Barbados, and several very talented students that have been through the lab, and also my colleagues at Granada and Navarra, who have worked with us for the metabolic reconstructions. Francisco and his group are experts on metabolic reconstruction networks, and Jose Angel and his group are experts in food sciences and helped us to interpret our food questionnaires and measure nutrients. And actually we're very happy because we just received some European funding to continue with this type of analysis, looking at the effect of specific foods on the behavior of metabolic networks. And that's it.